welcome students. Today we are going to study plant growth and development. So how do plants have different structure and functional attributes? You know that the plants have seeds, roots, stems, leaves, flowers and fruits. All this form a part of the plant growth cycle, in turn involving plant growth and development. Now the question arises, why do we need to study about plant growth and development? As we all know, we use plants in our day-to-day life for various applications, including food, medicine, paper, for textiles. We use plants for wood, used in constructions, and we use it as a feeding livestock. Now, in order to accomplish all these applications, a sound knowledge of plant growth and development is essential for assisting its healthy cultivation. That is the reason why we must know about the plant growth and development. So, in this chapter, we are going to study about in general the plant growth, what is the process of differentiation, dedifferentiation and redifferentiation in plants. Then in plant development, we will study about the role of plant growth regulators, photoperiodism, vernalization, and then how to overcome seed dormancy. So now what is plant growth? Now growth is a characteristic of a living organism, isn't it? So in order to define, it is an irreversible or permanent increase of a size of organ or its part or the individual cell. Now for this, it would require energy in the form of ATP, and it is accompanied by metabolic activity, which is a club of all anabolic and catabolic reactions. Now let us see, which of the following can be considered as plant growth? Can you people make out? Yes, the picture of the leaves and the plant shown on the right hand side of the slide shows expansion of the living matter, which is leaf, while on the left hand side it is swelling of wood piece when placed in water. So can you make out which will depict plant's growth? Yes, the expansion of leaf will be considered as the result of plant growth. As there is an increase in organ size due to metabolic activity which takes place with the help of ATP consumption, while in the swelling of the wood placed in the water, it happens due to the endoosmosis of water. So there is no metabolic reaction which is causing the swelling of wood. Now how do plants grow in nature? This growth in various different regions of the plant happens due to a set of cells which form a region called as meristem, and these places are called as meristematic regions of the plant, where there is rapid cell multiplication happening. So plants, they retain their capacity for unlimited growth throughout their life. That is a very interesting and a peculiar property which the plants have, that they can retain their property of unlimited growth till they are alive. This happens due to the presence of these meristems or meristematic regions in the plant. The cells in these regions, they are capable of continuous cell division and self-perpetuate. So as you can see on the picture, as you can see on the slide, the photograph shown is for the meristematic cells. So, meristem cells, they continue to divide to form new cells, and these new cells, then some of the cells, they differentiate to form parts of the bodies, plant body. So, this is called as open form of growth. Let us see what are the different types of meristems. So, if we can see on the slide here, there are black boxes encircling regions on the plant. These depict different types of meristematic regions present in the plant. At the top here of the plant, you see a region which is called as apical meristem. There is a microscopic picture of the section here where you can see the shoot apical meristem shown by the green arrow here, this region. It is found in the terminal bud of the plant, lengthening of the increase in the top region length of the plant. The bottommost region, the black box shown here, this depicts the root apical meristematic region or root apical meristem, which is responsible for the elongation of the root to go deeper into the soil. The third type of meristem is called as lateral meristem, shown in between here with a rectangular box. Now, lateral meristem is the region which is responsible for enhancing the width of the plant, which means the cork or the vascular cambium region, the increase in those regions. If you remember, the vascular cambium, it includes xylem and phloem elements, and the cork cambium is responsible for the growth in the bark region of the plant. So, these form the lateral meristems, and also the axillary meristem, which is responsible for branching to happen, showed here in the nodal region. So, this is another kind of meristem which is responsible for the formation of new branches. Let us talk about what are the different phases of growth which can be observed in plants. Now, just to give you an example, we will take root as the model system, and here we have demonstrated the various phases of growth in a root system. So, you can see at the tip is the region which is called as root cap, and as we go up the root, we see the regions of elongation with root hairs, which is a more matured region. So, let us talk about the bottommost region, which is marked here as root cap. This is the region which has meristematic cells. Now, these meristematic cells, they are rich in protoplasm. They possess large and clearly visible nuclei. The cell walls are thin and rich in cytoplasmic content. So, they are abundant plasmodesmata connections also, which you can find. Above the meristematic regions is the elongation region, where the cells, they will have increased vacuolation, the cells will be more enlarged, and the new cells will get, and there is new cell wall deposition. So, the cell wall becomes thicker. The yellow color region shown here with the root hairs is called as the maturation part of the growth, in which the cells will attain maximum size. There is no more division happening. The cell wall has thickened and there are protoplasmic modifications where you can see extensions in the form of root hairs. So, this is how growth happens in a root system. Now, the question arises, how do we measure plant growth? At a cellular level, you can see there is nucleus, cytoplasm, which combined is called as protoplasm. So, when there is growth happening, there is increase in the amount of protoplasm. Now, there are different parameters which are used to measure plant growth. Now, I hope you can understand that it is not possible practically to measure growth at the intracellular level all the time. So, there are indirect parameters which have been brought in to depict the plant growth. Like for example, plant growth can be depicted in terms of fresh weight or dry weight mass, or in terms of cell numbers, increase the rate at which the cell number is increasing, and if it is one dimensional, then we can measure the length of the plant, two dimensional area wise, and three dimensional volume wise. Now, mathematically there are two ways by which we can calculate the growth rate in plants. One is called as arithmetic growth. Now, the increased growth per unit time, this will be growth rate. So, anything rate means we are determining per unit time. So, growth rate would be the rate at which the growth is happening. Plant or plant's organ, they can produce more cells through arithmetic growth or geometric growth patterns, or in fact you can also observe them happening simultaneously. Now, let us understand what is arithmetic growth and under what conditions you can assume that it is arithmetic growth. Now, in mitotic cell division in plants, now one of the cells which has divided, which will again continue to mature and differentiate, while the other cell will continue to divide using mitotic division. So, if you can see on the slide, the two blue cells here, it will lead to daughter cell. One of the daughter cells is continuing to divide, while the other daughter cell will then keep maturing. So, this maturity happens because of its differentiation and gets added up to plant tissue or plant organ as per requirement. So, you can see, like for example in root elongation, there is a picture which shows the root growth in 10, 20, 30, 40 and 50 days interval. So, if we want to depict the rate of growth in this root, we can use the expression for arithmetic growth happening at a constant rate, where the length of the root at any given time t can then be given as the initial length of the root plus the rate at which the root is growing and the time, total time in which you want to determine the length. So, we can say that dl by dt, because the rate at which the length of the root is changing, we are assuming it is constant, which here it is being depicted by r. So, if you will integrate using the limits, let us assume at time t is equals to 0, it was at some initial value L naught, and at time t it is at some value L with a subscript t. So, if you will integrate, your RHS of the equation becomes L and it becomes R T and we have the limits 0 to T, and here the limit is L naught to L T. So, if you will put the limits, it will become L T minus L naught is equal to R times T minus 0. So, which is same as given here. Now, this is a linear equation. So, if you will make a plot as shown here in the yellow color of length of the elongated root, which is LT versus time, then it will be an inclined straight line with a y-intercept of L naught. I hope this is understood. And the slope of this line is nothing but your R, which is the rate of growth of the root, which is here assumed to be constant. As you can see from the inclined straight line, the slope is same. Now, let us see how we determine geometric growth. In plants, geometric cell division, it results if all the cells of the organism or the tissue are active and are continuously dividing via mitotic division. So, in that case what will happen? The 2 cells will lead to 4 cells, the 4 cells will lead to every cell leading to 2, so they will again lead to 8 cells, then these 8 will become 16 and so on. So, this is a geometrical growth pattern. Now, in geometrical growth pattern, one can see different phases of growth. If you will make a plot in terms of size or weight of the organ versus time, one can see 3 distinct phases of growth, which we call as the first phase, lag phase, where there is no significant change in the size or weight of the organ or the plant. Then comes the active growth phase, which is called as exponential phase. And then after the nutrient depletion happens, there is reduction in the growth rate and eventually there is no more growth happening. So, it follows more or less a sigmoidal growth pattern. Now, in the active growth phase, which is depicted here as an exponential phase, if we have to determine the growth rate of the plant or the organ or tissue, then we can use an exponential equation to find the growth rate. So, in that case, let us assume the y-axis is in terms of weight of the organ, depicted here as W1, and W0 is the initial weight. So, then with respect to time, this is the exponential function. The rate at which the weight of the plant is changing is proportional to the weight of the plant. So, mathematically, when this proportionality constant goes out, there is a constant which comes in, which has been given a notation small r, and we have W. So, if you want to make an equation with respect to W, we will integrate this as we did earlier. Let us see, put the limits here given as W naught initial value and at some time t they have given it as 1. So, let us put as 1 or t. So, time here initial is 0 and at some time t when the W is W1. So, if you will integrate this, you will get ln W and putting the limits W1 by W0 is equals to RT, t minus 0. So, we can say as RT. So, then writing it in terms of exponential function, the ln will go as, it is a base to the base of the natural logarithms. So, your W1 by W0 will become equal to e to the power of RT and W1 therefore becomes W0 multiplied by e to the power of RT as shown here. So, this is how we reach to the exponential growth expression for geometrical growth pattern in plants. You can even see this pattern in the seasonal variation observed in trees. When the leaves are falling, when there are fresh leaves coming and when the leaf buds are dormant during the winter season. So, you can also depict that change in terms of geometrical growth pattern. Now, we have come to know what is arithmetic growth and what is geometric growth. Here we have stated an example where one can see both arithmetic and geometrical growth pattern during plant growth and development. So, here the picture shows embryo of the bean seed, and during the embryo development phases, when it begins with the zygote divides using geometrical growth pattern, keeps dividing. It then slowly matures to a phase where not all cells keep dividing, but some of the cells they keep dividing, they retain division, while the other cells they tend to lose capacity to divide and they start maturing and differentiating to function various other roles in the embryo development until it becomes a seedling. Let us talk about absolute and relative growth rate. Now, what is absolute growth rate? It is the increase in the total growth of the plant per unit time, while relative growth would be growth of the given system per unit time, per unit the initial value of the parameter. Like for example, to understand it better, let us take this problem. There are two leaves as shown here, leaf 1 and leaf 2. Leaf 1 being smaller, initial size is 5 centimeter square. After 10 days, this small leaf reaches a size of 10 centimeter square, shown here in the form of dotted picture. Similarly, leaf 2 also in 10 days, initial size which is much bigger, which is 50 centimeter square. In 10 days, it increases to a size of 55 centimeters squares. Now, let us see how we can differentiate between the growth of the two leaves. If you see the absolute growth rate in the two leaves, it amounts to the same value. So, what is absolute growth rate? As I mentioned earlier, this is the increase in the area, in this case per unit time. So, the increase in area The difference is the same in both the cases, leaf 1 and leaf 2, which is 5, and this happened in 10 days. So, your absolute growth rate would be 0.5 centimeter squares per day, isn't it? As shown here. Now, if we want to compare the growth in the two leaves, because the initial size is different, absolute growth rate may not be the right way to make comparisons. Hence, in order to normalize the difference in the initial values, we make use of another parameter which I just mentioned, which is called as relative growth rate, which will take into account the variation in the initial values of the parameter. So, here we were depicting the change in the area to monitor the growth. So, now initial area being different in both the cases, if we use that also in the expression, we should be able to negate the effect or the difference in the initial values of the area. So, then the relative growth rate in this case can be written as the change in the area per unit initial area happening in the given amount of time. So, in both the cases, the difference in the final and the initial area is 5, while in one case the initial area is 5 and in the other case the initial area is 50. So, 5 by 5 divided by 10 days, and in the second case it will be 5 by 50 divided by 10, which is the time. So, here it becomes 0.1 and here it becomes 0.01. So, now you can see that in terms of relative growth rate, leaf 1 has a higher relative growth rate than the leaf 2. So, this can be used for making comparisons. Talking about the conditions for plant growth. What makes the leaves to increase in size? What facilitates this process of cell enlargement or the metabolic activity? Now, after reading the chapter on plant nutrition, we know that what are the basic requirements which lead to plant growth and development. Now, water, we know it will help in cell enlargement. It is also utilized in metabolic reactions, important for enzyme activities. Oxygen is a gaseous nutrient which helps in releasing metabolic energy. Light, again, it helps in generation of energy. Then nutrients, anyways these are required for synthesis of cellular content. Gravity, gravity is another parameters which is important, one of the parameters which is important for lengthening, determines the length of the plant and the length of the roots. Then optimum temperature, which is also very crucial for the growth of the plant, because it can impact the metabolism of the plant in terms of enzyme activities and the reaction rates. Now, what happens during plant growth in the cells? Plants, the change in the growth of the or the development of the plant is governed by three phenomena happening at the cellular level, which is differentiation, then dedifferentiation and again redifferentiation. Now, let us talk about these three processes. Differentiation, it is a process of maturation of cells which are derived from meristems to perform a specific function. So, meristems we know those are the regions where the cells continue to divide. So, they are actively dividing cells. Now, these cells they then undergo structural changes in the cell wall and their protoplasm. Like here in case of root or in the stems, you will find that some of these cells will mature to become the xylem elements. Here we have shown the vessel elements and the trichiads, which are different type of xylem elements required for the transport of water. So, dedifferentiation, what is it? It is the process by which differentiated cells again regain the capacity or the ability to divide. So, they return back to the ability to divide. So, when I was talking about differentiation, I said that the actively dividing cells, they stop dividing, they lose the capacity to divide and in the process mature to perform a specific function. Now, in case of dedifferentiation, a matured cell which is performing a specific function will revert back to a stage of meristematic activity, which means it will start to continuously divide. Like for example, the parenchyma cells. I am sure you must have heard about this term and you must have learnt about it. Now, these parenchyma cells, their major role in the plant is like storage or to become the flesh of the plant, the packing material. Now, these parenchyma cells, for example, they also dedifferentiate, revert back to meristematic state where to help in the intrafascicular cambium or in the cork cambium, and they facilitate in the secondary growth of the plant. In this case, it will increase the grit of the stem. So, this is a picture where you can see a cross section of the stem where the parenchyma cells, they have been modified to play a role in the interfascicular cambium and the cork cambium, thereby increasing the layers of the cells in the cork layer and also in the cambium region, the vascular cambium region here. So, vascular bundle is also shown here, the xylem elements shown in the pink. Now, talking about the third term, which was redifferentiation, process by which a dedifferentiated cells gain the ability to divide and perform the specific function. Now, once a cell has dedifferentiated, again it is redifferentiated to perform a new set of function. So, that is redifferentiation. So, vascular cambium, which is a dedifferentiated cells from the parenchyma cells, and they again get redifferentiated to form the secondary xylem or the phloem. So, this process can, the example which I quoted last in the last slide is a good example to show the conversion of or the process of differentiation, dedifferentiation and redifferentiation. Similarly, cork cambium, which is again a dedifferentiated cells, they will undergo the process of redifferentiation to form the cork, which is the protective or the waterproofing in the plant. So, this is how secondary growth enhancement will happen in a plant. You can see the picture here, the cork layer, which increases because of the cork cambium, and the secondary phloem and the secondary xylem, which enhances or increases due to the vesicular cambium present, and they form the wood of the plant. Talking about development process, it includes growth and differentiation. It is a term that includes all the changes that an organism goes through during its life cycle, from germination of the seed to its senescence. As you can see from the picture shown for sunflower plant, starting from the seed, then a seedling, a small plantlet growing to a mature plant, flowering of the plant, and then leading to senescence. Plasticity is defined as the ability of the plants to follow different pathways in response to the environment or phases of life to form different kinds of structures. A very good example which you can see in nature is the heterophily in coriander leaves. At the germination stage, you can see the leaf structure is different, while at maturity the leaf structure is completely different. It gets broadened. Another example in nature is the heterophily in buttercup leaves, where the leaves which are under the water, they have a thinner morphology, while which are exposed to the aerial region, aerial part, where they are exposed well to the sunlight, the leaf structure changes and they become broad and widen. Now let us see what are the factors that control plant development. There are intrinsic factors like the genetic makeup of the plant and the presence of certain components. Here abscisic acid is one of the plant growth regulators. Another is extrinsic factors, which includes light, temperature, water, oxygen, nutrition. So today we will discuss more about plant growth regulators. Now plant growth regulators, these are chemical messengers which are defined as organic substances which are synthesized in very less quantities in a part of the body, and they can be transported to another part of the body as per the requirement, where they would influence a specific physiological process. So there are various different kinds of plant growth regulators. We will study about the three classes: one auxins, another gibberellins, and cytokinins. These are plant growth promoting hormones or regulators. Talking about the regulators which are growth inhibitory, these are ethylene and abscisic acid. You must be wondering, why is the plant at all producing something which is inhibitory to its growth? But you will see later as we move on that how crucial it is for the plant to regulate its growth as well, and how does it help the plant. So talking about auxins now in little more detail. So there is a picture of a plant which has coleoptile regions, the tip of these plant leaves. It is canary grass which is shown here. Now they are found to elongate the cells of the tip region, which is coleoptiles, upon illumination. So when the light falls on it, one can see these cells elongating. This happens due to the presence of a natural auxin which is called as indole 3 acetic acid and indole butyric acid. So these are natural auxins which are present, which are said to be responsible for this elongation process. This can also happen using synthetic auxins, man-made auxins. For example, naphthalinic acid, acetic acid, naphthalene acetic acid and 2,4-D. So 2,4-D is a short form of 2,4-dichlorophenoxyacetic acid. Now these natural auxins, they are produced by the growing apices of the stems and the root. This can prevent fruit and leaf drop at an early stage. Auxins, they are also used to initiate rooting in the stem cuttings. You can see the picture on the slide here, where when the stem cuttings, they are inserted in a solution nutrient medium where auxins are provided, the rooting starts happening. They promote root formations. They are also used for or to induce parthenocarpy. You must have heard about seedless orange and fruits. So, they induce apical dominance, thereby reducing lateral growth, but the apical growth will happen. So, they will dominate. They are also used in agriculture in the form of herbicides. Let us talk about another class of plant growth promoting hormones, which is gibberellins. Now, gibberellic acid, this was first discovered in the fungal pathogen called as Gibberella fusiculae. They cause the disease called foolish disease of rice seedlings. Later, the same compound was detected in the plants. Now, gibberellic acid, it is used to increase in agriculture, to increase the length of the grape stalks. You can see the picture on the slide in the absence of the gibberellic acid, and when gibberellic acid is sprayed, then the wide difference which you can see in the length of the grape stalks. So, it hastens the maturity period, like for example from flowering to fruiting, so which is beneficial in agriculture. And it also delays senescence. So, can you guess what benefit can be achieved from this property in agriculture industry or in your fruits? Right, I hope you have guessed it. So, you can extend the market period of the fruits. It also promotes bolting. Here, an example in rosetta leaves. You can see the significant difference between the untreated plant and the treated plant, where lengthening of the internodal region just before flowering can be promoted. Let us talk about the third plant growth promoting hormones or regulators, which are called as cytokinins. Cytokinins, they are known to induce rapid cell division in plants. It was first discovered in the autoclaved sperm DNA from herring. Herring is the fish, and the kinetin was the first cytokinin which was discovered in its sperm DNA. Later, it was found to be produced in plants. Zeatin is another cytokinin which is identified in corn kernels and coconut milk. They are generally found near the root apices, shoot buds, and in the young fruits. You will find higher concentrations of cytokinins at these places in the plant. Contrary to auxins, cytokinins, they promote lateral growth. So, they will promote the growth of lateral buds, and therefore, they will suppress the apical bud dominance, and they also delay senescence by promoting nutrient mobilization. One of the other plant growth regulators is ethylene. Ethylene is used by the plants to promote senescence and abscission of plant organs, especially of the leaves and the flowers. It breaks the seed and the bud dormancy. It also promotes root growth. Ethylene is also highly effective in fruit ripening, as it enhances the respiration rate during ripening of the fruits. In deep water rice plant, one can see ethylene promoting rapid internode and petiole elongation. Ethylene can be made available to the plants exogenously through the precursors which is available commercially. It is ethophone. Growth inhibitors or regulators is abscisic acid. It was discovered to cause abscission of cotton buds or cotton balls. It inhibits seed germination, which can be a protective mechanism by the plant during adverse conditions. So, it will prevent sprouting during unfavorable conditions like desiccation. It helps by stimulating the closure of stomata in case of drought conditions, so thereby reducing the evaporation of water. So, it acts like plant growth inhibitor, which can be a response to any environmental stresses like cold temperature or dehydration. So, now I am pretty sure you must have made out that gibberellins, abscisic acid, these are antagonistic. Now, let us talk about a phenomena called photoperiodism in plants. The plants, in order to flower, they require certain day length, that is the relative length of day and night, which is called as photoperiod. Even when we do in vitro cultures of the plants or the plant tissues or cells, we ensure that the optimal photo period is maintained for maximum growth rates to happen in plants. So, the response of plants to the photo period expressed in the form of flowering is called as photoperiodism in nature. So, the shoot apices, they will modify themselves into flowering apices, but they cannot perceive photoperiods. So, the leaves which are present nearby, because they can perceive photoperiod, that is the site of perception of light dark duration, which then lead to a hormonal substance to migrate from the leaves to the shoot apices for inducing the flowering. So, this is how the plant regulates the flower bud maturation to flower. Now, depending on the nature of the photoperiod requirements, we can classify the plants in three types. One as short day plants, long day plants and day neutral plants. So, as you can make out from the term itself, the short day plants, for example wheat and green gram, their growth or their development, flowering, might be promoted when the light duration is less. However, if the light duration is increased or there are flash of lights during the night, the dark duration, then too you will observe that the development is affected in terms of flowering. In long day plants, like for example rice and radish, there is a longer duration of photo period which is required, and if the photo period or the light illumination period is less, then it may impact the flowering. Again, even in that non-illuminated period, if there can be flash of lights, that may also induce flowering. And in day neutral plants, no particular correlation is observed between the exposure to light duration and the induction of flowering response, like in case of sunflower or tomato. These are examples. There is another new term, vernalization, which refers to the promotion of flowering by a period of low temperature. So, let us see how vernalization is useful. In certain plant variety like winter variety of wheat, plant uses vernalization to complete its growth cycle. We know that the seeds are planted during autumn, and then when the winter arises, the seeds they mature into small seedlings. Now, during this cold period of growth of the plant from seedling and as it matures, the cold temperature effect causes enhancement in or induction in flowering, and therefore in spring season you see flowering happening, which then subsequently matures and is ready for harvest in the summer season. So, this low temperature is used by the plant to induce flowering in the growth cycle. The plants which follow biennial growth cycle, so even in the plants which follow biennial growth cycle, low temperature is used to bring about or to induce flowering in the plant and completing the growth cycle of the plant. So, the cold treatment in biennial growth cycle also induces flowering or photoperiodic flowering response. So, in seed dormancy, seeds which fail to germinate even when external conditions are favorable, in such cases the reasons could be immature embryo or presence of inhibitors like abscisic acid, phenolic acid or para-ascorbic acid, or the reason could be their impermeable hard seed coat. So, the measures which are taken to break the seed dormancy either involve removal of the seed barrier by mechanical aberration, either through knife or sandpaper, or aberration using biological agents like microbes, in which they are co-cultured or incubated for some time, which will bring about the rupture in the seed barrier, or removing the effect of the inhibitory chemicals. That is done by subjecting the seeds to chilling conditions, which we have learnt now called as vernalization, or by application of antagonistic compounds like gibberellic acids or nitrates, or changing the environmental conditions like that of light and temperature, the photoperiodic response. What we studied in plant growth development, it included studying the characteristics of plant growth, about meristems, the tissues which are responsible for the plant growth. We also discussed about the phases of plant growth, which included meristematic cells, elongation and maturation of the cells. Then we spoke about essential conditions needed for the plant growth. We discussed differentiation, dedifferentiation and redifferentiation processes. We spoke about factors that control the plant development, where we studied the effect of plant growth regulators and their functional role. We spoke about photoperiodism and vernalization, which is responsible in inducing flowering in the plants. Then we also studied about seed dormancy, about its causes and how to prevent. So, this brings us to the end of the chapter on plant growth and development.